Hello everyone, welcome back to the second session of Immunology class. And again, I am Dr. Sanmitra Ghosh from the Department of Biological Sciences, School of Life Science and Biotechnology, and we are from the Adamas University. So in our first session, we had a brief introduction about the humoral immune response, which is mediated by the immunoglobulins and the antibodies. And then we started our elaborate discussion about the structure and function of the antibody. So today in our second session, we are going to have an idea about the different types of antibodies or different types of immunoglobulins that we have in our body contributing to the humoral immune response. So let's start our studies today. For that, again, I'm going to share my screen with you. Yeah. So I have shown you this particular slide in our last session also. So we will begin it from here with the names of the different types of antibodies which are normally present in the body of the human. And here they have been categorized based on their relative amount in the circulation of a normal healthy individual. So we have the IgG, IgA, IgM, IgD, and IgE. And today also we will be learning in details about these immunoglobulins according to this particular chronological manner. So first of all, we will start with the most abundant form of antibody which is present in our body, and that is IgG or immunoglobulin G. Now, before starting about this IgG, I would like to mention here one very important point regarding this classification of the immunoglobulins. See, here we have given the names G, A, M, D, and E to different immunoglobulins. They have the basic same structure. That means there are two light chains and two heavy chains. But the difference is there that here the heavy chains that are present are different. Light chains, they can be of two different types, the lambda and the kappa. And in all these different types of antibodies that we are going to learn, either lambda or kappa light chains are present. Now, based on the type of heavy chain that is present or the polypeptide chain that is present, the names of the immunoglobulins are decided. So, these heavy chains, it can be of 5 different types. If gamma heavy chain is present, then it will be the immunoglobulin G. If alpha heavy chain is present, then it will be referred to as immunoglobulin A. If the heavy chain polypeptide is mu, then it will be the IgM. If it is the delta polypeptide chain, we will be having the immunoglobulin D. And if it is the epsilon polypeptide heavy chain, then it will be IgE. So, students, you can understand that there are 5 different types of heavy chain possible. That is gamma, alpha, mu, delta, and epsilon. And these polypeptide chains also, they are having the abundance in our system in the particular chronological order that I mentioned. That is gamma, alpha, mu, delta, epsilon. So, as we are starting with IgG, so here we will be having the light chain as either lambda or kappa. But the gamma chain will always be, or the heavy chain, better to say, will always be gamma. So, IgG, this is the a very short picture, very small picture of the, and a very simple version also of the IgG structure. And it is the most abundant antibody, and it is mainly present as a monomer in our system. This IgG is also referred to as maternal antibody because this is the antibody which we inherit from our mother as we are born. So every newborn child, when he or she is coming out of the mother's womb, he or she is coming with a basic innate immunity, and the maternal antibodies, some of the maternal antibodies are carried forward to the child, so that when the child is immediately exposed to the external environment, that maternal antibody will give it protection against the invading microorganism to a certain extent, till its own antibody producing mechanism has become activated, has become optimal. Okay. And this particular maternal antibody that is coming against, it can be polio, it can be diphtheria, it can be any other form of disease, but their nature is that they are IgG in nature. So among these 5 different types of antibodies that we are going to learn, this IgG is the only antibody which can cross the placenta and provide the immunity to the child, which can last for 1 to 2 months after birth. And within this 1 to 2 months, as it is continuously facing the assaults in the external environment, its immune system is getting trained and it is now becoming ready to produce the antibodies of their own. Now, IgG, it has the molecular weight of 1,50,000 Dalton, where the heavy chain is 50 kilodalton each, and the light chain is the 25 kilodalton each. For IgG, there can be 4 different subtypes based on the variation in the amino acid sequence in the polypeptide chain. For the heavy chain, it can be of 4 different types. And since the amino acid sequence is different, so the folding is also different, and which can also affect their functionality. So there is a subtle difference in the functioning of these 4 different subtypes of the IgG. So as I mentioned, it consists of 2 gamma heavy chain and 2 kappa or 2 lambda light chain. Here one thing you must remember, it is never the combination of 1 kappa and 1 lambda chain. It is always either 2 kappa chain or 2 lambda chain. Now, of the 4 different types of IgG that we have, IgG1, IgG3 and IgG4, they are much more efficient in crossing the placenta, and they have the most prominent role in giving protection to the fetus that is developing in the mother's womb. Ig3, among all these 4 different types, Ig3 is the most potent activator of the complement system, which is followed by IgG1. On the other hand, IgG2 or IgG4, they are not that much involved in the complement activation. Now again, this IgG1 and IgG3, they can bind with great specificity, with high affinity to the receptor of the Fc region on the phagocytic cell, and they can mediate the opsonization. IgG4 and IgG2, they, in case of opsonization as well as for the complement activation, they play the minimal role. So as you can see from this stratification of their functionality, IgG1 and IgG3, they are playing the most important role. And IgG is the form of the antibody that is secreted first from the B lymphocytes. So in case of the B lymphocytes, we have seen that either the antibody is present on the membrane, bound to the membrane, or they are being secreted out from the effector cell or the plasma cell. So the secreted version of the antibody that we are getting from the plasma cells, they are IgG in nature. But the membrane-bound antibodies which are present on the surface of the B cell, they are not IgG in nature. They are IgM in nature, and we will be studying about them later. Right. So now we will move towards the second most abundant form of antibody, and that is the IgA. Now, its structure, as it has been shown over here, is considerably different from that of the IgG. Because in case of IgG, it was monomeric in nature. And as you can see over here, IgA, this is not monomeric, but it is at least dimeric in nature. Now, this second most abundant form of antibody, it is consisting of the alpha heavy chain polypeptide. So again, here the light chain can be either lambda or kappa, but the heavy chain will always be alpha. Okay. Now, this IgA, it is found mainly in different types of body fluids. What kind of body fluids? It can be the mucosal secretion from the mucous membrane. It can be the breast milk of the mother. It can be the tears. It can be the saliva. So they are present in the external secretion also. So that is why it is also referred to as the secretory antibody. So which one is the secretory antibody of the human body? It is IgA. Now, all of the mucosal surfaces, okay, that is on the surface of the mucous membrane, where we have the MALT. What is MALT? MALT is the mucosal associated lymphoid tissue. So this can produce the IgA. So whatever mucous membrane we have in our body, there we have this lymphoid tissue where from this IgA is generated. Now, initially when it is formed inside our bone marrow, we all know that in case of the B cell proliferation, they are getting activated in the bone marrow. So when these immunoglobulins, they are being formed, they are actually present in our blood circulation. Okay. Later on, some of the IgA, they are secreted out and then they are referred to as the secretory IgA. So please note that IgA is never formed inbuilt as the secretory IgA. They are later on converted to the secretory form. So that means they have some kind of modification in their structure to become the secretory IgA. So what is that modification? So initially when the IgA is being encoded or it is being synthesized, it is synthesized in the form of a monomer, just as we have seen in case of IgG. But when they are modified and they have been designated as secretory IgA, at that time it consists of a dimer of two IgA molecules which are covalently linked by a polypeptide chain, and that polypeptide chain is the J chain. So if we look into this particular picture, then this Y-shaped structure is the one of the monomer and this is the second monomer, and here we have this dimeric structure which is linked by this yellow block, which is the polypeptide chain, and that is the joining region or the joining chain or the J chain. Now, in the serum, primarily, when this IgA has been produced, as I mentioned before also, they are produced in the form of monomer. But the polymeric form, here we have shown the picture of the dimer, but it can be dimer, trimer, and sometimes tetramer formation. It is also observed in case of the IgA. So, multimeric form of the secreted version of IgA has been noticed. Okay. So, the polymeric forms, whenever these polymeric forms are being formed, they are consisting of the J chain polypeptide. So, in the serum, J chain polypeptide is not there, but in the secretory version, always when we are having the multimeric structure, this J chain polypeptide is always present. Okay. Now, the IgA of the external secretion consists of a dimer or tetramer. First of all, a dimer or tetramer, then a J chain polypeptide, and apart from that, there is an additional polypeptide chain which is known as the secretory component. So, mainly in the external secretion or the secretory IgA, there are four components present. That is, we will be having two antibodies, that is the dimer of the antibody or a tetramer of the antibody. Then we will be having the J chain joining those antibodies, and there will be an additional polypeptide chain, which is known as the secretory component. Now, where is this secretory component coming from? The secretory component is derived from the receptor, that is the poly-Ig receptor, which is responsible for the transport of the polymeric IgA across the cell membrane. So the receptor on which this IgA is being bound, from that particular receptor itself, a portion of the polypeptide chain is being cleaved off, and that is forming the part of the secretory component. Okay. And what is the function of the J chain? J chain actually helps in the polymerization of the serum IgA as well as the secretory IgA. And one of the very important feature of the secretory component is that it will mask the sites of the Fc region, particularly in case of the IgA, which are susceptible to protease cleavage in the hinge region of the secretory IgA. And this allows the polymeric molecule to exist for a longer span of time in the protease-rich mucosal environment, which otherwise would not have been possible. Because as I have mentioned before, that from all the mucosal surfaces, this IgA can be secreted. And since in the mucous environment, huge amount of protease activity is observed. So any kind of protease that is coming, they can cleave at the particular recognition site in the hinge region. And if the antibody gets cleaved off, then there is no chance of secretion of this antibody in the external secretion. So to prevent that, to prevent the conversion of the functional antibody to a non-functional one, this secretory component actually masks those particular region of the Fc region where those protease recognition sites may be present. And as they are masked by this secretory component, this protease, they cannot cleave over there, and so the antibody remains in the functionally active form and they are secreted. In the next slide, we are going to have the pictorial representation of what I say just now. That is how the antibodies, they are actually, this IgA antibody actually, they are secreted out. So, as you can see that here we are having a plasma cell derived from the precursor B cell, which is now dedicated for secretion of the IgA. Okay. So from this protein secretory or protein synthesis machinery, we have got the IgA molecules, IgA, multiple number of IgA molecules are there. And also since the J chain is also another polypeptide chain that can also be derived from this protein synthesis machine. So they are secreted out from the B cell. So when they are secreted out from the B cell into the submucosa, the dimer of the IgA will be formed. And once this dimer of the IgA is being formed with the help of this J chain in between, they will migrate towards the lining of the cell membranes. And there we have the poly-Ig receptor. So the dimeric structure bound by this J chain will attach to this poly-Ig receptor. And once it has attached over here by the process of receptor-mediated endocytosis, so here this poly-IgG is the receptor. Once the antibodies bind to it, there will be the endocytosis mediated by the poly-Ig receptor. So this entire complex will be internalized. Okay. So once it has been internalized, so a vesicle has been formed, and inside this vesicle, different lytic enzymes are present. Okay. So this lytic enzyme, they will now cleave this poly-Ig receptor to release a portion of it. And that particular portion is referred to as the secretory component. And as you can see that this particular vesicle will now merge with the other side of the membrane, and on the lumen side, the secretory IgA will be released. And how does it look like, and how is it different from the previous one? Here we have this dimeric secretory IgA linked by the J chain, and the Fc region of both the components of the dimer being masked by the secretory component. So, this is the method by which the secretory component is released, bound to the dimeric IgA, constituting this entire complex constitutes the secretory IgA in the external fluid. This is the third most abundant antibody, and that is the IgM. And as we have seen in case of IgG and IgA, here also you can understand that in case of IgM, the heavy chain that is present will be different, and it is the heavy chain mu. If mu is present, then the antibody that will be derived is IgM. The most vital point about IgM is that they are the first type of antibody that have arisen due to the evolution. That is why when a B cell is formed with the surface of it, outer surface of it studded with the B cell receptor, which are actually membrane-bound immunoglobulins, those membrane-bound immunoglobulins are actually IgM in nature. Later on, when the B cell becomes converted to the plasma cell, then the secreted versions that we are getting, they are IgG. But evolutionarily, they are the first type of antibodies which are produced, and that is IgM. As you can see from this picture here, how many monomers we have? 1, 2, 3, 4, 5. So 5 monomers are present. So these 5 monomeric units are polymerized together, and they are linked over here with another J chain similar to that of the IgA. So IgM is a polymer of 5 monomeric units which are arranged in a pinwheel array. As you can see over here, in a pinwheel array which are held together by the J chain. So as I mentioned, this is the first antibody made during the B lymphocyte maturation, and they have low affinity and very high avidity. Now, what is meant by this affinity and avidity? Affinity means it is the strength of the non-covalent interaction between the antigenic determinant, that is the epitope, and the hypervariable region of the antibody in the variable region that corresponds to the complementarity determining region. So, when one antigen is interacting with an antibody, the number of non-covalent interactions that is holding them together, the strength of it, that is referred to as the affinity. Now, in case of IgM compared to IgG, the affinity is lesser. That is because in case of IgG, we had the hinge region present. So, IgG was a molecule which consisted of 3 C regions in the Fc zone. Okay, 3 sub-C regions were present. And because of the presence of the hinge region, now what we have in the hinge region? In the hinge region, we have the presence of proline amino acids. And because of the structure of the proline, that gives flexibility to the structure of the antibody. So if this is the antibody, if my hand represents the antibody, and in the hinge region we have proline, so here as you can see, we can move these antibodies so that it can accommodate the antigen properly, so that the antigen binding becomes optimum. Now in case of IgM, since this hinge region is missing and the constant region is divided into 4 segments, C1, C2, C3 and C4, at that time the movement of the antibody over here in at the end of the Fab region, here it is not maximum. So the binding of the or the insertion of the antigen and interaction of the antigen with the Fab arm, that is restricted. It is not as flexible as the IgG. So since here the movement of the arm is restricted, that's why here the affinity is not as high as that of IgG. And next important component is the avidity. Now avidity means it is the sum total of the amount of non-covalent interaction that is holding the total number of antigens with which the antibody is interacting with. Now, since IgG or IgA, they are monomeric or dimeric in nature, so the number of antigens with which they can interact with their two Fab arms is limited. But in case of IgM, we have how many arms? 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. So large number of antigens can come and bind with a single molecule of antibody of IgM. So if we take the sum total of non-covalent interaction between the total number of antigens with this IgM, that will be much more higher in case of IgM because it is having this pentameric structure with 10 hands ready to hold different antigens. So that is why IgM is said to have much more higher avidity, that is the strength of binding, total strength of binding of the antigen, that is much more higher compared to that of the IgG. So these 5 monomeric units, they are held together by the disulfide bond that link their carboxyl terminal heavy chain domain, particularly in the C4 and the C3 region. And as they are having that pentameric structure, there are 10 different antigen binding sites. So their avidity is higher because they are having a greater valency for binding different antigens at the same time. And this pentameric IgM is also much more efficient than its other isotypes. Other isotypes means IgG or IgA or IgD or IgE. It is much more efficient in binding the antigens with many overlapping epitopes or many repetitive epitopes, as we can see in case of the viral cells or in case of the erythrocyte. IgM is also more efficient than IgG in activating the complements because there the avidity is much more high. So on the surface of the bacterial cell, it can interact with multiple antigens, and that brings a change in greater avidity helps to bring the change in the structure of the C1 component, that is the initial component of the complement activation, and this process becomes much more efficient over there. And IgM lastly plays an important accessory role as a secretory immunoglobulin. So apart from IgA, IgM can also be a secretory component, but the abundance in case of secretion, the abundance of IgA is much more higher than that of IgM. Our next candidate is the IgD. And IgD is normally present in very low concentration in the blood, and they are monomeric in nature with the structure almost similar to that of IgG. So the difference between IgG and IgD will be that in case of IgG, we had the gamma heavy chain, and here we have the delta heavy chain. And they are susceptible to proteolytic enzymatic activity as well as they are sensitive to heat. But another very important difference between the monomeric form of IgD and IgG is that IgD cannot cross the placenta. So we cannot inherit this IgD from our maternal end. They are abundantly found on the memory cells. So once from the antigenically committed mother B cell, we are getting the memory B cells and the plasma B cells. In case of the memory B cells, on the surface, the BCR which are present, or the B cell receptor which are present, they are normally IgD in nature, and they also play a very important role in secondary antibody response. Last but not the least, we have the IgE, and this IgE is present in extremely small amount, and in some individuals it is not present at all. So it is present in the blood serum either scantily or it is completely absent, and it varies from one person to another. They are also monomeric in nature, but since they are associated with the allergic reaction, with the hypersensitivity reaction, they are also known as the allergic antibodies, and they can mediate the immediate type of hypersensitivity, particularly type 1 hypersensitivity reaction. Now, in case of IgE, just like IgM, we have no hinge region. So another extra constant domain is present, that is up to C4 we have the constant region domain, which helps IgE to bind with the specific receptor on the mast cell. So as this IgE can bind with the IgE receptor, which is referred to as Fc epsilon R1 or Fc epsilon R2. Two such receptors are present on the surface of the mast cell. So there, when more than one IgE molecule comes and binds over there, and they interact with the multivalent antigen, it can be any pollen grain, it can be any food molecule, it can be any venom, it can be any toxin, and that particular antigen binds and cross-links at least 2 or more IgE antibodies. A single IgE antibody can never trigger the process of hypersensitivity. At least 2 molecules are needed. Sometimes more than 2 molecules can also participate in triggering this allergic reaction. So when this cross-linkage actually forms, that leads to a change in the signal transduction inside the cell, and as a result of which, in the mast cell, we have different types of vesicles or pouches which consist of pro-inflammatory mediators. So once this interaction has occurred from the mast cell, we have the degranulation. So based on this degranulation, from the granules which are present in the mast cell, different pro-inflammatory mediators are released. And these pro-inflammatory mediators, they are actually responsible for the different manifestations of the allergic reaction. It can be anaphylaxis, it can be hay fever, it can be sneezing, it can be other types of inflammatory responses, and they are because of the different types of pro-inflammatory mediators which are released from the mast cell, and they are characteristic feature of the allergic reaction mediated by IgE. So these are the different types of antibodies that we have in our body. Apart from this categorization, there is another way of categorizing the antibodies or the immunoglobulins, and they are known as the isotype, allotype, and the idiotype. So in our next class, we will be studying about this isotype, idiotype, and the allotype. Hope you will enjoy, and see you soon.